Hello, everyone. My name is Michael Muscus, and I'm a segment marketing manager here at ATCC. We're so glad you've joined us for the next installment of ATCC's Excellence in Research webinar series entitled Yarrowia lipolytica, a versatile, non-convention oleaginous yeast for biotechnology, presented by Professor James Krill from Syracuse University and Dr. Scott Wynn from ATCC. In today's presentation, you'll learn how Yarrowia has transformed from a notorious butter and margarine spoilage organism into a cutting-edge platform for industrial biotechnology. Professor Krill teaches microbial forensics and biochemical analysis while conducting research on Yarrowia genomics and biomanufacturing at Syracuse University. His work focuses on comparative genomics, strain engineering, and forensic applications of microbial systems. Dr. Wynn is a microbiologist bioinformatician who has described several microbial species and identified multiple SARS-CoV-2 variants. He holds a PhD in microbiology and immunology and has previously worked at USDA, University College Dublin, and the DC Public Health Laboratory. Today, Professor Krill and Dr. Wynn will discuss how Yarrowia has evolved from a contaminant to a versatile biotech platform, offering unique traits for biofuel, oleochemical, and protein production. They'll show recent research highlights, including sequencing of 38 strains, the first phase diploid assembly, and pangenomic insights that reveal structural variations and new opportunities for industrial biomanufacturing. During or after the webinar, if you have any questions for our speakers, please use the chat function available through the webinar program. All questions will be answered as allowed at the end of the presentation. And as a reminder, the recorded webinar presentation will be archived on the ATCC website at www.atcc.org. Also, feel free to click on the links provided in the webinar console. You can connect with our speakers via LinkedIn or email. They're excited to connect with you. Now, before we begin the webinar, I wanted to share some information about ATCC. ATCC is a nonprofit organization founded in 1925. We are headquartered in Manassas, Virginia, with additional facilities in Gaithersburg, Maryland. ATCC is a leading global supplier of highly authenticated biological materials. We're a strong collaborator within the scientific community, and we're an innovator of solutions to enable efficiencies in life science research. So with that, I'd like to welcome Professor Krill and Dr. Wynn.
Hi, thanks, Michael, for the introduction. I wanted to start off with telling you a little bit about Yarrowia lipolytica and its interesting origin story that actually illustrates how industrial problems can drive scientific discovery. So back in the early 1900s, butter and margarine manufacturers had a major contamination problem, and Yarrowia was the culprit. So Laura Rogers, who actually founded ATCC, was studying butter spoilage when he characterized this organism. So Yarrowia has literally been in ATCC's DNA from the beginning. The taxonomic history is messy as it's been renamed multiple times, which actually causes some confusion in the literature even today. But what's interesting is what made it a nightmare for margarine industry is exactly what makes it valuable for biotechnology now. Then Jacobson at Unilever figured out that controlling this yeast was the key to stable margarine production. So industry interest in this organism actually goes back for over a century. So the historically problem organism or problem child status actually reveals something important about its biology. What's remarkable about Yarrowia is that even though it was first considered a contaminant, its characteristics are beneficial from a biotech perspective. So its lipolytic enzymes that spoil dairy products are the same ones that make it really good at breaking down waste oils and fats. Its environmental tolerance to low pH, high salt, oxidative stress, and it can even grow on hydrophobic substrates where most other yeasts wouldn't survive. So, this resilience means you can run industrial fermentations on crude or contaminated feedstocks because it thrives in harsh conditions that other microbes might not be able to handle. These unique capabilities position Yarrowia differently than conventional yeast platforms. When we look at industrial yeast platforms, Yarrowia checks almost every box that you might want. So your 30% lipid content in wild type strains is already impressive. But there have been engineered strains that go beyond 50% lipid content. And the applications span biofuels, chemicals like biodegradable lubricants, high value proteins. So essentially, if you're working with lipids, fats, oils, or unusual carbon sources, Yarrowia should be your first choice, probably not your backup. But these biotechnology capabilities don't evolve in the lab. They came from where and how Yarrowia lives in nature. So understanding its ecology helps explain its industrial success. So examining where Yarrowia lives in nature helps explain why it works so well in industrial settings. It's globally distributed, being found in soil, seawater, dairy facilities, wastewater treatment plants, basically anywhere there are lipids that are degradable. This is what initially attracted me to Yarrowia. It's actually an indicator species for identifying different stages in the post-mortem interval. There's growing interest in using it for bioremediation, like cleaning up oil spills or industrial waste, because it naturally survives in these harsh environments. So the ecological diversity has driven something fascinating at the strain level. There's remarkable variation in how different isolates look and behave. One of the most striking things about working with Yarrowia strains is how different they are. If you look at these colony morphologies, you'll see two extremes, one being very dry and white on the left to very oily and opaque on the right. These are all the same species, but with dramatically different phenotypes. So this variation extends beyond just the appearance. Different strains have distinct lipid profiles, substrate preferences, stress tolerances, and even growth rates. So this suggests to us there's been niche specialization within the species. So some strains adapted for dairy environments or marine or soil habitats, for example. So for strain engineering, this is actually advantageous. You can screen across strains to find the best starting chassis for your specific application rather than forcing everything through one reference strain. This phenotypic diversity we can see raises an important question. What does the genome look like? This brings us to the type strain sequencing project that we'll discuss later in the talk. But for now, I'd like to hand it off to Scott.
Thanks, Jamie, for introducing me. I want to take over and talk more about the biotech applications of Yarrowia lipolytica. So Jamie provided some details on his use not his use, but its background as a contaminant in the butter and margarine industries, and also as an indicator marker for cadavers. So I want to switch tracks to look at the biotech applications of Yarrowia. So Yarrowia lipolytica has real industrial history. In the 1950s, British Petroleum explored Yarrowia lipolytica for its biotech application, including the single-cell protein. So they wanted to look at Yarrowia and its ability to grow in hydrocarbons on N-alkanes. And N-alkanes is a byproduct of petroleum distillates, hence the interest by BP. They developed a commercial protein product called Toprina G, and there were many studies on it as a use for a protein-rich animal feed. They did studies in lambs and on chickens and found that these organisms grow really well on single-cell proteins. However, in the 70s, BP was constructing a plant in Italy before the Italian government actually suspended permission to market Toprina G. This, and combined with the petroleum crisis in 1973, led BP to ultimately withdraw from the single-cell production market. However, it's essentially an early sustainability story, but later on, other commercial companies built upon its success. So recently, in the last several years, a regulator asked the European Food Safety Authority to have this panel on nutrition, novel foods, and food allergens to look at Yarrowia. And they found that Yarrowia, as a novel food, is actually considered to be safe, and it's the use of this biomass when it's dried and heat killed is not nutritionally disadvantageous. So it's actually safe for use, and this marketer was looking for its usage as a food supplement. And on the right side here is a image of this Toprina G product that was produced by BP. So fast forward to today, the current state of research in Yarrowia, it has a mature genetic toolbox. There's a lot of tools at its disposal now, and this organism has demonstrated industrial readiness, including commercial scale fermentation. So we have CRISPR-Cas9 Golden Gate assemblies, and also it's by pairing these tools with whole genome sequencing, researchers can actually manipulate this organism much better now. In addition to the European Food Safety approving the use of Yarrowia for human usage, the organism is also considered generally recognized as safe by the FDA. So recently, in the last several years, DSM, which is a Dutch food company, put an application to use Yarrowia lipolytica as a genetic engineered platform for the production of Reb-A. Reb-A is a modified steviol that's derived from stevia, and it is much sweeter and less bitter than other steviol compounds. And this Reb-A is grown in Yarrowia lipolytica. And in this report, Yarrowia is considered to be a BSL-1 and not to produce toxins. And in 2015, the generally recognized as safe status, or GRAS, was approved by the FDA in 2015. So Yarrowia has a promising and already proven track record as a chassis for recombinant proteins, and also for production of like single cell oils or citric acids or other compounds. It's also very versatile in its ability to use waste oils, glycerol, other waste products. And thanks to whole genome sequencing and these tools, the pathways for lipid biosynthesis and organic acid production is well characterized in this organism. So for molecular tools for Yarrowia, engineering has evolved in phases. So in early research, back when BP was looking at Yarrowia, UV radiation and breeding Yarrowia isolates were some of the tools used to manipulate Yarrowia. However, in the early 1990s, we had gene knockouts such as Cre-Lox-P for marker recycling, and also Ura3 blaster. Later on, programmable nucleases like talons were introduced for targeted knockouts. However, they were limited by the efficiency, and also is very labor intensive. Recently, with the CRISPR-Cas revolution, CRISPR-Cas9 is the efficient platform for genome editing, and it allows you to actually multiplex edit the organism. So multiple genes can be knocked out. And CRISPR AI has been used for gene repression and activation as well. Multiple examples here, like the TRP1 knockouts or PEX10 knockouts, illustrates that people are using these tools to knock out multiple loci to tune pathways and not just do single knockouts. And it's not just editing. These genome-wide CRISPR libraries can be used for screening along with promoter swapping in Yarrowia. So it's a very versatile organism for this research. This is where Yarrowia starts to look like a true chassis. So there's a lot of domestication that's been going on in Yarrowia. A lot of these oxytrophic markers like UR3, LU2 have been knocked out in certain strains, and this allows researchers to really control how the organism behaves. There's also some protease deletions. So for protein stability, so knocking out these proteases allows Yarrowia to grow a protein target of choice without having it degraded. There's also the regulatory systems that matters for production. So inducible promoters like erythritol inducible systems help manage burden and phase production. And with the chassis development themes, you can have boost precursors for acetyl-CoA, NADPH, and they optimize pathways for lipids and terpenoid production, and then the use of CRISPR-Cas9 for genome precision control. So now we pivot to what ATC brings for you as a researcher. So we have a number of strains in the collection, and not just that, a number of Yarrowia have been sequenced for the ATC genome portal. These were sequenced through a hybrid approach with short and long reads. Many of these strains are actively used in biotech research. So you'll notice that PL1F there, which is in the middle, is a descendant of another widely used Yarrowia strain. And PL1F actually has several genes knocked out and is currently used in a lot of commercial applications. A key point we want to emphasize is that all of these strains in the AGP are sequenced the same way using both short read and long read. And they're also assembled with the same pipelines and annotated with the same tools. So this actually increased consistency with genomic data. Another thing to note is that Yarrowia assemblies on the AGP are more contiguous. There's less contigs than the ones in NCBI. So if your workflow depends on accurate gene models, that difference shows up immediately in your design phase. And finally, all of the Yarrowia genomes in the AGP are fully annotated. When you look at NCBI, only 14 Yarrowia genomes are actually annotated. So we provide a much more consistent platform for researchers. I want to bring focus to the new taxonomy tree in the ATCC genome portal. You can easily search the genome portal with a taxonomy tree. It's quite interactive. You can search for various strains across the portal, and you can also request any strains that we haven't sequenced yet through this feature as well. And it's quite integrated. You can see it's formally organized under taxonomic ranks in the collection at ATCC. So one thing that we hinted at earlier is that the assemblies in the AGP is much better than the ones you find in the NCBI. So there is a reference assembly of Yarrowia lipolytica in NCBI. It's a strain of Yarrowia known as W29 or CLIB89. We also have this strain. It's known in our collection as ATCC 20460. And in the manuscript talking about the W29 reference for NCBI, researchers mentioned that there was a 54 kb repeat in their assembly of CLIB89. However, in our assembly of CLIB89, you do not see this 54 kb. And when we map reads to this region, we found that there's only a single copy of this 54 kilobase region. And this repeat that's found in the NCBI RefSeq might have resulted from either a misassembly or potentially a laboratory adapted variant of W29. Another thing we wanted to point out is the LU2 gene in Yarrowia lipolytica. So whenever these researchers uploaded the RefSeq assembly for W29, they also mentioned in the manuscript that this is the first instance of the LU2 gene in Yarrowia being sequenced. When we were comparing the LU2 genes on our assembly versus the W29 reference, we found there were a number of differences between the translated W29 LU2 gene and also the ATCC 2460 LU2 gene. We found there's differences in the C terminal, and whenever you blast the W29 LU2, it only matches itself in CBI. However, if you blast LU2 from ATCC 2460, it matches other Yarrowia proteins from other laboratories in NCBI, and also matches the other Yarrowia sequences we have in our genome portal. So as a sanity check, we matched our sequencing reads and mapped it to the ATCC 2460 assembly along with the W29 reference gene. And we found that our sequencing reads confirm what we suspected, that our assembly of LU2 is correctly assembled versus the one that's, again, found in NCBI. And again, that could be potentially of a misassembly in the NCBI reference or a lab-adapted variant of W29 from that research laboratory. And from here, I can pass this back on to Jamie on our publication on type strain genome.
All right, thanks again, Scott. That was great. So now we're going to talk a little bit about the type strain project that we collaborated on. And this type strain genomic assembly represents a major milestone, not just for Yarrowia research, but for how we think about reference genomes. So this is the first phase diploid assembly of 18942, which is the official type strain for the species. This means we separated both copies of each chromosome and can see heterozygosity. This matters because most assemblies tend to collapse diploid genomes into single representation where you lose structural variants and heterozygous alleles. So for metabolic engineering, knowing which genes are heterozygous, which have allele-specific expression, and which structural variants exist is going to be critical for rational design. So having this high quality reference enabled us to compare structural organization across strains at the chromosome level. When we aligned the type strain against the reference W29, we found something interesting. Despite it having greater than 99.5% sequence similarity, the chromosome structures were very different. We're seeing reciprocal terminal translocations, with the largest being about 1.4 megabases. These aren't small rearrangements. And since the transposons didn't explain these rearrangements, we decided to dig a little bit deeper into the transposable elements in order to understand what role they did play. The transposable element landscape gave us some insight into Yarrowia's evolutionary history. We identified diverse transposable element families. The annotation identifies most of these as relics, which is a classification the pipeline assigns to degenerate or fragmented copies. Those historical transposable element artifacts, before they became inactive, likely contribute to the chromosomal rearrangement that we observe between the strains today. And these transposable elements and their patterns could potentially offer some forensic value that is of interest to me. So if the transposable element insertions vary between strains, they might serve as molecular fingerprints for identification. So given that this is a globally distributed organism, we might be able to use this information for attribution or identifying where something came from. So far, we've seen structural differences between two strains, but how do they compare at the sequence level across the entire species? When we compared 70 genomes that were collected from ATCC, NCBI, and NREL, we found that Yarrowia is both highly conserved and structurally diverse. So the FASTANI on the right calculates how similar genomes are by comparing their individual DNA sequences. So the heat map shows all pairwise comparisons where the yellow means they're nearly identical and the blue means that they're a little bit more divergent. So this shows all strains fall within a really tight species boundary. There's no cryptic speciation or deep divergences despite the global sampling of all of these strains. However, there's clear phylogenetic clades. This isn't just random variation, there's actual population structure here. This could suggest that the gene sequences are highly transferable between strains, but chromosomal rearrangement and regulatory differences will affect how these genes actually perform. So then we asked, what is the full genetic repertoire of the species? Do different strains carry unique gene sets, or is the metabolic toolkit essentially fixed? The pangenome architecture that we see in this Anvio plot tells us about the evolutionary strategy of Yarrowia. So the Anvio will show which genes are shared versus unique. The black core contains genes represented in all strains, and the outer rings show genes present in only some strains or multiple copies of some genes in individual strains. So that large core genome means almost all core functions are conserved. There's not a lot of horizontal gene transfer bringing in novel capabilities. The accessory genome suggests that the pangenome is moderately closed. So the implications for genetic engineering is that the metabolic toolkit may be largely fixed. So optimization may come from tuning, regulation, and expression. The good news is that this stability is advantageous for industrial applications. So with these genomic insights, from structural plasticity to pangenome stability that we've discussed throughout this talk, all point to why having a high-quality sequence assembly and authenticated reference strains from ATCC is essential for reproducible research. And I'll hand it back to Scott to wrap up our segment.
Thanks, Jamie. And to conclude from this webinar, you see that Yarrowia lipolytica has a history as a contaminant, but now has multiple applications in the biotechnology. In the biotechnology usage, has an exceptional metabolic flexibility and the ability to accumulate a lot of lipid, which underpins the value of Yarrowia lipolytica. Even though Yarrowia lipolytica has a moderately closed pangenome, there's enough diversity to have biological significance and potential applications for biotech usage. The ATCC has a number of high quality curated genomes along with the type strain, and this genomic data paired with the strains in the collection are essential for reproducing engineering and also for the tracking of strains. And we also provide reliable strain selection. There's a number of Yarrowia lipolytica in the collection that we're constantly sequencing, and it allows for the data we have allows for comparative genomics and also the ability to translate this genetic data to industry. So I want to acknowledge not just our partnership with Syracuse University with Jamie Krill, but also our partnerships with Capra Biosciences, researchers from Virginia Tech on the uses of Yarrowia lipolytica and its metabolic diversity. So far, we have a publication, as Jamie alluded to earlier, is the complete phase diploid assembly of the type strain of Yarrowia. Various team members from ATCC has contributed to this project. Can't really list them all, but you can see their names there. And also, I think for Syracuse, Jamie, do you want to talk about them?
Yeah, I'd like to thank everybody at Syracuse, as well as everyone else on the other teams that have helped with this project and the future projects going forward.